Brilliant. No, thanks again, Laura, and thanks to the Pharmacology Society. It's a real delight to be with you all today, and thank you all for taking the time. I know it's quite late on a Wednesday, so I appreciate it, and hopefully it will be of some benefit, or at least time well spent. So I've called this talk "The Winding Road of a Scientific Career." And the reason why I describe it as a winding road is because things are never straight. Things are never go down a straight path. And also, as you will see from my narration of my career, it wasn't necessarily something that I had explicitly planned out in any great detail. And I'm very much a believer of this notion of sensing and responding. So planning, but not too much. So I'd like to give you just some perspectives from my own journey. This will be a personal perspective. In biotech and pharma, I've spent over 15 years in those sectors. I am a very strong and sort of core scientist that has ventured into these arenas and has had the privilege of doing a variety of things. And I'd love to share that with you today. So perhaps just to start with, I thought it might be useful just to give a brief introduction beyond what Lara has mentioned on my life, essentially in a few bullets. So I'm a father of 3 young energetic boys. One is a speed Rubik's Cuber. You can check out his YouTube channel, YH Cuber. The other has a mean right foot, and he's not on YouTube yet. And the youngest is 2, and so is really keeping us fit, especially in this quarantine and lockdown era. I'm a scientist with an academic background in immunology and T-cell biology, and a deep belief in harnessing the power of the immune system to treat deadly diseases. I've worked in large pharma and biotech, and have been privileged really to oversee the research and development process from target discovery through to early phase clinical trials in melanoma patients, and also have been privileged to steer organizational growth. As Lara mentioned, I'm currently CEO of a small Norwegian biotech developing cell therapies for solid cancers, and I'll talk a bit more about that later. Really, my passion is about lifting others, and I'm very inspired by translating science into impact for patients, and really doing this by unleashing human potential. So that's, if you like, me in some bullets. What I thought I'll spend some time on today is a few things. One is my own journey, some of the questions I had along the way and the decisions that I made, and I'll just maybe take you through the thought process for me. Of course, everybody's journey will be different, but I thought it may be helpful to take you through mine in case that sets a framework for how one may think about, or at least set some universal principles that may be helpful. I'd also like to give some reflections on scientific and management career paths. This is a conundrum for scientists, especially those that move into the healthcare sector, into pharma or biotech. It can become a question, and some find it difficult to grapple. Also, I'd like to give some reflections on biotech and pharma from my own perspective and my own experience, and perhaps growth areas and final thoughts to end up with. So really, when I embarked on what do I do, all the way back in 1996, it's as you all know, not a straightforward question. And the way I had looked at it for myself was, I had asked myself and introspected on what do I enjoy. And really the big themes, the 3 themes, and some of you will resonate to this, is helping others, applying science to the discovery and implementation of medicines, and making an impact. Those were 3 of the key drivers or guiding principles really behind my initial choice for embarking on a journey and a degree in biotechnology at UCL. So that really was the start for me of my discovery and curiosity into deeper science. And what I've done in the next few slides is I've tried to mark key milestones in my own pathway. And I've tried to give some of the key questions that I've asked along the way and elements that have marked this period for me. So some of the things that really stood out for me during this period. So with my degree at UCL, I did a degree in biotechnology. It was a fairly new degree at the time. Really, the elements that marked that period for me were the scientific depth, so the exploration of science as part of a degree. The industrial process, biotechnology as a degree also offered further depth into understanding industrial process, which was something different compared to some of the other life sciences degrees like biochemistry or molecular biology or straight biology, for example. But also the offer with this particular degree was a placement year to be really organized by the individual, so in this case by myself, in a pharma company or other organization. And I would say that was incredibly influential. And I was lucky enough to land a placement with GlaxoSmithKline, and I worked there for a year as a sandwich placement just after the second year of the degree until I went back to complete my third year of the degree. So that provided real-world experience and really began to set my curiosity for a further depth of understanding into science. And so after the degree, I asked myself, what do I do next? And really the placement at GlaxoSmithKline triggered my decision to pursue a PhD. And the reason for that is that it was twofold. One was my passion to really understand a particular area in depth. And the other was the benefit that I had seen in having gone through a PhD or seeing others that have gone through a PhD and then applying themselves in industry. And that was the exposure that I had at GSK and seeing that for myself firsthand. And so in making that decision, the next fork in the road, as it were, is what do I study further and where? And of course, Oxford is well renowned for the life sciences and really highly regarded in terms of the quality of the science. And I managed to find a position within Neil Barclay's group at the Cellular Immunology Unit. And the Cellular Immunology Unit is really cutting edge when it comes to dissecting the role of the immune system to combat various diseases. And so I decided to do that. And I would say the elements that really marked this period for me, as shown in the bottom, are the independent research, critical thinking, both of which come with, I think, going through a PhD, the resilience that one needs to actually get through a PhD. I recall in my first few months, I was spending time actually running a basic PCR, which took some time to get right, but also required some resilience to continue with. And then, of course, I, during that period, developed a depth of knowledge in immunology, technical expertise in cell biology, and that really began to kindle a passion to harness the immune system to combat diseases. And I would say this is the period really that kindled my passion to really apply the immune system in its entirety, or harness the immune system to combat diseases. And so the next question beyond the PhD was, what do I do next? I knew that I had and I felt a strong desire to directly apply my scientific learnings to the discovery and development of treatments. And because I was privileged to have the support from my previously established GSK relationships, which I had established during my industrial placement, that really facilitated my first professional positions. And so one of the themes I'd like to, one of the messages that I'd like to relay as part of this talk is relationships. Relationships really matter. Um, the impression one leaves really matters, and staying connected really matters. And so the irony is that I actually went back almost to the same seat that I occupied when I was doing my sandwich placement many years earlier. So I went back to the same department, and it was an incredibly enriching experience at GlaxoSmithKline. And I would say the elements that marked that period for me were the professional discipline in research and development, the infrastructure, the resources that were available to enable research and development in a variety of different indications. It was also working in what's called a matrix environment, so working with multidisciplinary teams, being part of a larger project with people that represent different disciplines, whether it's development, whether it's biochemists, whether it's biologists, whether it's bioinformaticians. Project management was also another component that was developed in my time at GSK. And also the exposure to the research and development process, particularly the early stage process. I was very much focused on the early stage process in the department that I was in. And so I was very much focused on early stage discovery to hit identification. It was also an arena to be exposed to different biology, which is linked to different disease areas, oncology, so cancer, inflammatory diseases, respiratory diseases, all of which are disease areas that many large pharma have activities in. And I would say the period through GSK initiated or started my transition to leading people. Over that period, I had transitioned from an investigative scientist to a group leader with a small group that was tasked with doing a specific task for the department. In this case, it was to develop assays that could be used for the screening of particular candidate molecules that may be relevant to certain disease indications. So after that period, there came a point where I began to start reflecting. And some of those reflections are shown at the top here. So the now what. And this wasn't, I wouldn't say this was an active now what. So this wasn't a strong urge to do something different. But there was a number of simmering desires. One of those was to move to a smaller biotech. I had been appealed by the notion of actually working in a biotech with the kind of the entrepreneurial spirit of a biotech, the agility of a biotech, the potential to get direct exposure to broader research and development processes, and actually to be closer to patient application. And that was one of the strongest desires. I also had a strong belief in the potential of harnessing the power of the immune system to treat cancers. If you recall, that was something that was kindled really during my PhD. And I could see that the field was moving in the direction of immunotherapy at the time. And so bearing in mind, this was 2009, and this wasn't really at the time where any of the immunotherapeutic breakthroughs had gained ground. That was perhaps 2011 and beyond. But certainly there was a move to really trying to develop more and more immunotherapies or find ways to harness the immune system to combat diseases. And I could see that there was some movement in that direction. And so an opportunity emerged for a biotech that actually brought together many of those elements, perhaps all of those elements, with a company called Immunocore, which is not far from Oxford. It certainly taps into the talents of Oxford. It's in Abingdon. A small biotech at the time, 40 people at the time. I was the 41st employee. I think I was the first from industry, actually. It was mainly and mostly academics. And I joined and started in the biology department. And I would say that period of my professional career really was enriched and enriching for me, and a real privilege, and I believe a unique experience to gain the exposure right across the R&D breadth of process, from target validation all the way through to early phase clinical trials. And so the elements for me that marked this period was the full transition to a leadership and management role, gaining experience contributing and overseeing the full breadth of the R&D process. At one point, I was privileged to be overseeing target validation all the way through to early phase clinical trials in the setting of metastatic melanoma, where we were treating patients, melanoma patients, with our first-in-class molecules. Immunocore is a company that specializes in developing what are called T-cell receptor bispecific therapeutics, and so those were being tested for the first time. The other element that marked this period were the relationships. Relationships, as I mentioned, are very important. Organizational growth. The company grew from 40 people to 450 people when I left, and so I was again privileged to be part of that growth phase. And also, it allowed me to explore next stage organizational practices. I'm very passionate about how people can come together and really do extraordinary things, and have really delved into looking at next stage organizational practices. How can one bring together groups of people to really empower them, and more than empower them, but allow them to and enable them and serve them to really reach their highest potential individually and collectively. And so I had the opportunity to do that also in this period. The other aspects that come to a certain extent with biotech are the exposure to the different elements. So the fundraising, I had responsibility there, strategy, science and business strategy, business development as part of the growth of the organization, intellectual property, which is very important, particularly for a small organization that's based on a protected idea. And over that period, I built an expertise in immuno-oncology, so the application of the immune system, or harnessing the immune system to treat solid cancers, and also the same to treat infectious diseases. And really for me, I would say the most profound element is the first direct impact on patient lives. I remember distinctly when I was asked to lead the first in human clinical study. At one point, we received data from one of the clinicians, and actually this was a clinician that we were working with in Oxford, and they had just scanned one of the patients from our trial, which had shown substantial shrinkage of the tumor. And I remember taking the scan to the chief scientific officer at the time, Ben Jacobson, who's a great friend and mentor for me, and just being incredibly excited with him about the response in patients, which marked the first response. So the first direct impact on patient life really was another element that marked this period. And really, after that, after almost spending 9.5 years, almost 10 years doing a variety of different things with various responsibilities, an opportunity arose in a small biotech with some intriguing science and a need to build. And there's a company called Zelluna Immunotherapy, which is where I currently am. It's a Norwegian-based company, actually only runs out of Norway. So I'm currently talking to you from my home in the UK. And an opportunity arose again in an area that is of great interest for me, so in immunotherapy, specifically in cell therapy, and also working in a specific area that I have built an understanding on, T-cell receptors, and really from an organizational perspective gave the opportunity to build again. And if you recall, Immunocore had grown to quite a substantial size. I had realized as I reflected on my own areas of interest that I very much enjoyed the building phase. So I joined Zelluna in 2018 as CSO. It's a very small company, joined at the time, and we were around 10. Currently, we're around 12. We're 12 in the company, and transitioned to CEO at the start of this year. And really, the elements that mark this period so far are the intriguing science. It's a really novel area of science, which is embedded in an area of science that I have worked on and worked with for over 15 years. For me, it brings together my prior experience in science, in R&D, in working with people, building teams, the network that I have been lucky to build over the years, and also organizations. And so it's been a real privilege to work with Zelluna and to really build on the scientific idea and really set in motion the cultural direction with the team and start to break through in that space. So that's a bit of a whistle-stop tour of my own career with some of the questions that I had asked myself, some of the elements that really mark this period, and hopefully that begins to give an understanding of perhaps some of the really important themes that, for me at least, string together the different aspects of the various parts of my career. And so the next couple of slides are around some of the choices and considerations. And again, these are my own thoughts around this, and these are also reflections based on my experience, really. And I would say, as one embarks on, as scientists embark on a track, on a career track, and if that's in an industry, so in biotech or pharma, there may come a point where a decision will need to be made around how one wants to develop their career, whether that's lab-based completely, or whether that's leading people, or whether that's somewhere in between. And some thoughts on my own experience here, and as you can see in the picture on the left, it is a true picture of me. I was trying to help out in a bit of a situation that we had in Zelluna, and I would say, I take every opportunity to go to the lab. Actually, it's really a lost passion for me. Probably best that I'm kept out of the lab these days, but it's certainly something that I enjoyed and found joy in, which I think is difficult to compensate for. So certainly with a lab-based role, one gets the elation of scientific experimentation and results generation. The first person to actually see that data coming off the machine is probably a joy that's difficult to compensate. You maintain connectedness to technicalities, and that's by virtue of doing the lab work, and of course the proximity to scientific direction. Of course, when leading people, there is the opportunity to shape the culture of a group, to really unleash the potential in people, to preserve them and allow them to reach their greatest potential. And of course, through others, one can have potentially quite a broad impact. And as we think about some of the elements of success in each of these types of pathways, I'd like to just raise a few of those, and these are not mutually exclusive. There will be some overlap here, but just certainly for me, some of the things that perhaps are highlighted in each of these tracks. For the lab-based role, of course, the rigor, the independence of working and following through the experiments, and scientific leadership that one shows will stand them in good stead for a successful lab-based career. Communication of data is fundamental, the ability to communicate data very clearly to all sorts of audiences. Integrating data sets across disciplines in relation to projects. So this is really trying to bring together data sets, not only ones that one is generating themselves, but also bringing that together with other data sets that are emerging for a particular project, and the ability to integrate that and bring that together and find the meaning behind those outcomes, again, will stand a lab-based career person in good stead. Record keeping is fundamental, particularly in industry. It's of paramount importance, and keeping abreast of the field, constantly keeping abreast of the field's specific area of expertise, but also perhaps beyond that too. When it comes to leading people, I think elements of success undoubtedly include passion to serve and give time to people. Ultimately, it's about the people when one is managing or leading people. It also requires decision-making in the context of ambiguity and uncertainty, and there is much ambiguity and uncertainty in industry, in pharma and biotech, and leaders would need to make decisions and be comfortable with making those decisions by looking at the balance of the risk and benefits and also the unknown information. They also, what will stand leaders in good stead is the ability to bring clarity and prioritize, and be able to bring that clarity to the group or to the department or to the organization, and also to prioritize appropriately with focus, and the focus here on really what's sometimes called the bigger picture or the higher why for the organization, and really move them towards this higher purpose. Again, communication of data is critical, and connecting the dots and strategic thinking become more and more important as you lead people in an organization. So these are just some reflections around lab-based and or management or leadership roles within organization, and some of the things that they could provide in terms of elements of excitement, and some of the things and elements that would stand one in good stead to really thrive in those careers. In terms of biotech and pharma, I get asked this question often, and sometimes by interviewee candidates. And I'd just like to summarize, and this is not probably not new to the group here, but just again, reflections from my own perspective. And if we run through biotech first, one of the motivations for me was the clear line of sight to clinical application. And I recall when I joined Immunocore, which was my move from GlaxoSmithKline to Immunocore, 1 month into my role, I was out with a delegation from Immunocore to speak to the FDA, which are the regulatory body in the US, and talk to them about the potential application of one of our therapeutics to patients. So really getting closer to clinical application. And at the time I was representing biology in those discussions. And I remember clearly saying to myself that I felt my choice to move to biotech was really vindicated in that moment. Biotech typically very focused on a technology or a project or an idea, and indeed that's how they tend to be formed. Smaller companies, they tend to be smaller companies, so you are exposed to most activities and actually can contribute to many. Resources are typically scarce in biotech, and so application of one's own initiative, curiosity, talent to activities outside of their own area is typically welcome in a biotech, and particularly if there's an aptitude for learning. They can be very dynamic, fast, high pressure, which also means potentially high stress, and a very vibrant environment. With pharma, you're exposed to a variety of projects, as I was, and a range of therapeutic areas. You have a matrix working environment, so it really brings you and introduces you to a way of working that brings together teams, each of which is representing a different discipline, the so-called matrix. The operations are very professional. Of course, they've been established for many years, and they have established infrastructure. And so there's good practices around how to project manage, how to carry out investigations, and really solid infrastructure that for me provided a wonderful training ground actually in a professional environment. And for me, was hugely beneficial as I then moved to a biotech, where I could then deploy some of these practices that I had learned at GSK. The availability of resources are greater typically, and that's financial, but it's also people resources. One can tap into the expertise of people across the organization, thousands and thousands of people if it's a large pharma, for example, and you can really draw into that expertise. With a biotech, one can do that, but that's typically outside of the organization if it cannot be found within the organization. And also, as I mentioned, in biotech, resources are typically very scarce. Some elements of success with biotech is really very much rolling up your sleeves and getting involved. Thriving in uncertainty. There's a lot of ambiguity and uncertainty, particularly in biotech. A growth mindset, if those of you are familiar with Caroline Dweck's work, growth mindset, and really an aptitude for learning outside of one's expert areas. I mentioned that typically is welcome because resource is scarce. So if one is curious and has an aptitude and would like to apply themselves outside of their own area, so if they're actually within the research organization but are interested in project management, or are interested in intellectual property, or are interested in business development, then applying themselves to those areas is typically welcome, especially if there's an aptitude for that. In terms of pharma, working in a matrix environment and being able to do that well, building relationships across departments is important and a key element of success, and also influencing others. That becomes very important when working in a pharma organization and where one wants to develop and really thrive in that organization. And really with both organizations, to various or to a greater or lesser extent, initiative, decisiveness, particularly in biotech, flexibility, communication, rigor, and amenable to change. Change can happen, it does happen, and so being flexible and amenable to change and really embracing the change, understanding what it means, and really trying to find a path forward in the context of that change is another element of success. I would also say never underestimate what people call the soft skills, and perhaps rather than what do you want to be, I have more and more been interested in the question of who do you want to become. And I think that's because really the skills that one develops by introspecting and thinking about who they want to become, around communication, persuasiveness, rhetoric, around empathy, flexibility, around aptitude and learning and growth mindset, and around the relationships that they have as part of the person they become. I think those are very, very important things to think about, and of course can be developed and can be habitualized and reflected on really at any time. So what do senior biotech leaders do? These are just some of the elements of what senior biotech leaders do. Provide a vision for the company, unleash the potential of those they serve. That's a critical element. As Aristotle put it, the whole is greater than the sum of its parts. So I spend a lot of time trying to find how I can remove any barriers from those that I serve in the company and allow them to reach their full potential individually and also collectively with others. To set the cultural direction of the company. For us at Zelluna, we're very much focused on what's called a deliberately developmental organization, constantly seeking to learn from each other, but also from outside. To identify growth areas for the organization, to support financing of the company, and to support operations. So what helps? It helps to have a passion for people, an ability to communicate a compelling vision, something perhaps that others can't see for themselves. Focus and clarity is critical and what really matters. And experience in various aspects of research, development, and clinical process, and a strong scientific understanding. One of the things that I feel was very important for me was the exposure that I had, particularly through Immunocore, of not only research, but development and clinical and contributing and actually leading in those areas, which I feel has helped to allow me to do what I can as a scientific leader, understanding what it takes in those arenas and also contributing to operations and being able to ask, hopefully, the right questions. Experience in business development and intellectual property, again, something that can come with biotech, an area that is very crucial to biotech, and something that can come given the small size of the biotech organization. So inevitably something that one would be exposed to. So what 4 things helped in my own journey? Relationships, and just a practical tip here: establish relationships, maintain those relationships, perhaps send a courtesy note every so often to those you're connected with. Maybe there's an article you've come across that they may be interested in. And, you know, we're now in the era which we weren't in my time. We're now in the era of LinkedIn, which makes all of that very easy. One thing I say is plan, but not too much. I do believe in sensing and responding. So sensing how the situation is at the time, what your change in motivations may be, and how you see the field moving. So perhaps a practical tip here is be clear about what you want to achieve or develop in the next year or two, but also be receptive to new information. Commitment, and for that, there's nothing can beat doing something you really believe it. That's been one of the core drivers for me. And exposure and contribution to the breadth of research and development and early clinical phase process. So really try to get exposure to the breadth of the entire process and seeking opportunities for that. If one is in a company, for example, setting, one can do that by spending time in different departments. And that's usually welcome in companies, particularly, as I mentioned, in a small organization where that application and that resource, as it were, would be very welcome. So in terms of pay scale and sort of financial reward, there's an approximate pay scale, and these are based on my own experience. Coming in as an associate scientist, you'll be looking at that range as shown here, up to 29,000. Scientists with a PhD would probably be in that range as shown in the table there. Senior scientist, again in that range. And then one starts to think about leadership roles or technical expertise. So then the pathway can start to diverge into principal scientist or group leader, technical expert, department leader, CSO or VP if you're in a pharma, and then CEO or SVP if you're in a biotech or pharma. So those are the kind of ranges. So as you can see, it can also provide some financial rewards as one develops in their career. In terms of the growth of the field, immunotherapy, which is something that I focused on, is a really growing field. I'm sure many of you know about it, have heard about it, perhaps are within the space. But as you can see from the various front covers that emerged a number of years ago, it really is a breakthrough space. Probably got going about over 100 years ago with William Coley's toxins, and finally in the last decade we're seeing real progress and revolutionary, I would say, responses with immunotherapies applied to cancers. And one of the areas in that space, the so-called cell therapies, is an area that I am currently working in with Zelluna. And this is a schematic just to demonstrate what cell therapy is. It's essentially, and has been thought, at least the first wave of cell therapies have been T cells that have been engineered to target cancers, either engineered to have a guidance system that's based on chimeric antigen receptors, so antibody-based guidance systems shown at the top here, or T cell receptor guidance systems, which are the natural way that T cells actually recognize diseased cells, such as cancer cells. And what we have seen with these types of therapies is really remarkable response rates. really outstanding response rates, and in some cases cures for patients treated with these types of what are called engineered cell therapies. So what do we do at Zelluna? Well, we work in the cell therapy space. I mentioned we're a privately held company in Oslo, established in 2016. We develop cell therapy products for solid tumors. We have really a unique approach where we use T-cell receptors and we put those into natural killer cells. Natural killer cells have numerous advantages when it comes to attacking cancers. They're naturally cytolytic. They naturally have a disposition to kill. But they also are useful because they don't cause graft versus host disease. So my natural killer cells, for example, can be used with those of you on this webinar without causing graft versus host disease. So what that means is that one can take a single donor, generate a bulk a source of material and doses from that donor and treat multiple patients. It's really a privilege to work with an experienced cell therapy team, all the way from research through to clinical development. This is all of us actually in the building that we occupy, the Oslo Cancer Cluster. 12 of us. Actually, it's missing one of the new researchers that we hired recently, but it's a small but really powerful team. And we've established a number of partnerships that we believe will help us to really deliver on our vision to treat patients with novel, what we call TCR-NK therapies. And just to give you a flavor of the kind of responses that one can see with cell therapies in general in the field, the first wave of cell therapies were what are called CAR T cell therapies. I talked about the chimeric antigen receptor, the CAR T cells, and really what you can see are response rates of 83%, 63% complete response in liquid tumors. And companies like Novartis and Kite have these on the market. These are marketed agents now, and they're treating leukemia patients with very high response rates. We're also seeing companies that are local to Oxford, Adaptimmune, for example, which are probably the leaders in T-cell receptor T-cell therapies. This is a scan from one of their data sets showing that across different diseases, so liver cancer patient, skin cancer patient, gastroendometrial junction patient, you could see there's tumor shrinkages when you compare baseline scan to post-treatment, where they have seen tumor shrinkages across a variety of different indications. I call these the first wave. So these are very specific cell therapies. So a patient comes in and their cells are taken out. They're engineered to contain a new T-cell receptor or a CAR, and then that material is put back into the same patient. That's what's called autologous T-cell therapy. The second wave is what's called off-the-shelf cell therapy, and this is the area that we're in. This is where one can take a single donor, engineer the cells, but then introduce those cells into multiple patients. So not just to a single patient, but to multiple patients. Of course, that offers the potential of scalability. It offers the potential to really treat and impact more patients. And really, I won't go into this slide, but really the responses that are starting to emerge with this off-the-shelf type cell therapy, and natural killer cells can be used as off-the-shelf, as I mentioned previously. One donor can be used for multiple patients, is demonstrated in this report recently, where liquid cancer patients were treated with CAR NK cells, again with very high response rates, and so starts to build data around the potential of these off-the-shelf therapies, particularly those that are based on natural killer cells. So what we do is we take T-cell receptors, we put them into natural killer cells, and we give them an additional function to their natural killer innate function, a function that allows them to target cancers that are expressing the target for the T-cell receptor that we put into the natural killer cells. And for the interest of time, I'll skip the next slide. We have a publication on this. It's shown at the bottom here, which you're welcome to have a look at to demonstrate the concept. But it really is a high activity and a high investment space. Organizations and investors are wanting to really harness the potential and the real effects that we have seen with cell therapies, but try to take that to a new level where one is using off-the-shelf cell therapy. So really bring together the effectiveness of cell therapies, but try to make that more scalable with off-the-shelf cell therapies. And so you can see there's a lot of deal making and investment that is going in, and that's really expanding. So very much a growth area within the cell therapy space. So finally, and just to save time for some questions, I thought I'd share with you a few responses I gave to in an interview recently, which I thought were quite penetrating questions. The best advice I was given, activity is not always progress. We tend to find ourselves being very busy, and that's the nature of the world we live in, and sometimes we can get caught up in busyness and convince ourselves that being busy means we're making progress. But actually, many times it doesn't. So it's worth taking a moment to think about where one is and whether all of the activities are really activities that are allowing one to progress. Advice I would give, I tend not to give advice. I think everyone has their own sort of influences and journeys, and difficult to really give advice to people. But what I would say is plan, but not too much. I've mentioned that before. I think your passions, motivations, and interests evolve over time, so send in response to these. You know, there are many elements of my own career which I didn't plan. What I wish I'd known, trust the journey. It sometimes does require you to look back to understand the wisdom behind the path you've trodden. I sometimes talk about my own career in 3 segments. I learned about T cells. I then learned about how to professionally develop drugs. And then I went back to use T cells as drugs. So really connected the dots for me. And my simple philosophy in life is I like to borrow from one of my favorite philosophers, Lao Tzu, who says, when no credit is taken, accomplishment endures. Really doing things for doing its sake, really, rather than for any acknowledgement, really. So with that, I'll thank you for listening. Hopefully there was perhaps 1 or 2 things that may have been of interest. And I'd like to just leave it open now for any questions. Lara, is it over to you?
Sorry, yes, thank you, Namir. Thank you for a great talk. I think your consideration and perspective are great guidance for all of us. And it's great also to hear from someone that is so passionate and about the impact that your research is also having in the field. We have so many questions coming, which is great. We'll try to ask the most voted questions. And the first one is if you would recommend doing a postdoc before applying for a position in the pharmaceutical industry.
Yeah, I think, you know, these are all very personal choices, of course. I think the benefit of a postdoc, from my perspective, the benefit of the postdoc is that, it continues to provide further depth in an area of expertise, which can be very helpful as you enter into the pharmaceutical industry. I would say I would caution, if there is a desire really to move into industry, I would caution a long stretch in multiple. I would caution multiple postdocs. I think there is an opening for those that have really developed deep expertise in certain areas of industry. So not to say there wouldn't be an opening, but I think one does potentially reduce the options if you delve very deeply into academia. So I think a postdoc or 2 can add merit if one would then want to move into industry, but it could become limiting to a certain extent if it goes beyond that.
Okay, great, thank you. There's a question if you could speak about a part-time PhD program and how convincing teams to have these programs for 7-8 years. Is maybe your company doing something similar, or?
Yeah, so we don't have a program like that. There are industry sponsored PhD programs, and they can take a number of years. I think the merit of those is having a foot in the door, as it were. You know, is really getting the exposure into industry, building the relationships. I think, you know, this is very much all about relationships. It's about people seeing what you're about, what you're passionate about, how you engage. So I think there is merit in those, even if they do take longer than a fully academic PhD. We don't run those at Zelluna, unfortunately.
Okay. Is there something similar for postdoc, like an industrial postdoc?
There is. So Genentech are very sort of prominent in that, and they believe in trying to bring in postdocs, because one thing that industry is really trying to do is trying to not lose the creativity, and really try to take the creativity from academia and bring the practices of industry and bring the two things together. And so you hear people like Vas from Novartis talking about unleashing the creativity, reimagining medicine. And so organizations and companies are you know, advocating such things. Genentech is one of those for postdocs, for example. So they do exist. They're not very pervasive, but they do exist.
Okay, great. The next question is about medical doctors. How can they use, 1 second, sorry. If they're more interested in preclinical and translational medicine rather than later stages of clinical trials, how can they best use their medical doctor in this for this advantage? And
yes, and that again, that's a personal choice, and that depends on the area of interest. I think that, you know, for me personally, what I'm most passionate about, if I was to really distill it to one thing, is the translation. Is that translational space. So the early phase 1, really testing something in patients and learning from them. And for that, it requires a certain sort of passionate clinician, and you know, an early phase clinician, but it's an incredibly enriching phase. But that may not be, you know, something that clinicians would engage with. They may prefer the more established phase 2, phase 3 trials, but certainly there are opportunities across that entire spectrum. I personally would say it's very exciting to be in that early phase trial, and I know many clinicians and worked with quite a few that I've worked with actually on the Immunocore and also in Zelluna's organization and on Zelluna's program. So it depends. It's a personal choice, but there are opportunities, and actually they're difficult to find really good clinicians, particularly for the early phase. So, you know, if one develops experience there, if they develop their kind of expertise in that space, they'll be certainly very well sought after.
Okay, great to hear. There is opportunity for them there.
Absolutely, yeah.
This is a very nice question. I think we could end with this one. Is asking, what do you wish you knew when you were our age? So I guess at the very beginning of your career.
Yeah, I think that, you know, I would say, again, you know, it might not sound so profound, but really to trust the journey. I originally applied to do medicine, and it's not something that happened. But I look back now, and I think that you know, I'm grateful for the journey I've had and the impact I've been able to be privileged to have on patients. And I spent a lot of time and stress, I would say, in just wondering where my journey will take me. So I think if one can sort of minimize that stress and that burden of thinking, where will I end up? And really just plan for the next 1 or 2 years, be guided by what they enjoy, and make connections and establish relationships. You know, I'd be happy to speak with anyone. I've got my details here if I can help with anything. But really, you know, try to liberate themselves, just to take it at one step at a time. And I think, you know, when you look back, you'll really see the wisdom behind it all.
Thank you. It's such a great advice to hear in these stressful times for all of us students and your research career. Thank you. I think we can end the talk. It's almost 6, so everyone can enjoy their evening. And we'd like to thank you again. It was really, really helpful with all your insights on this talk. And yes, thank you again for joining us, and I hope to good luck for your company as well.
Thank you so much, and thanks for organizing, and happy to stay in touch.
Thank you.
Thanks, Lara. Take care. Have a good evening.